Good afternoon, everyone. So this is the session for the oral presentation finalist 3. And my name is Alina, and I'll be your moderator for this session. With me, we have two very distinguished judges, Prof. Dr. Wayne Hodgson and Associate Professor Dr. Amoda Kadivelu. So just a bit of housekeeping. I've asked all judges and presenters to keep their video off and their mics muted until the end of the presentation, where I will invite the judges to ask a question to the presenter, and the respective presenter can on their mic and answer the question promptly. Okay, so let's start. We have 6 presenters altogether today, and our first presenter is Amirul Faez Shamsudin from University Science Malaysia.
Bismillahirrahmanirrahim. Assalamualaikum. My name is Amirul Faez bin Shamsudin, and the title for my oral presentation is Investigation of the Associations between Genetic Polymorphisms and Cholesterol-Lowering Effect of Statin among outpatient statin users in hospital USM. So let's move on to introduction. So what is statin? Statin is commonly used as medication to reduce cholesterol levels. And mechanism of action of statin takes place inside the liver during cholesterol biosynthesis process, in which statin inhibits conversion of HMG-CoA into mevalonate, therefore causing reduced production of cholesterol in the liver. So how does statin affect lipid profiles in plasma? Focusing on atherogenic lipoprotein, statin treatment able to reduce level of atherogenic lipoprotein due to increased uptake to the liver. While for non-atherogenic lipoprotein, statin treatment able to increase the level of HDL due to reduced transfer of cholesterol from HDL to atherogenic lipoprotein. Statin efficacy in reducing cholesterol may vary in different persons, and this varying efficacy may due to several factors which include genetic polymorphisms. This figure summarizes the gene that involved in statin disposition as well as statin efficacy and statin side effects. Now, talking about genetic polymorphisms, single nucleotide polymorphism is the most common type of genetic polymorphisms. And based on candidate gene approach, 8 SNPs within 7 genes have been selected for this association study, which is listed in the table below. Therefore, we hypothesize that these selected genetic polymorphisms are associated with cholesterol-lowering effect of statin, and our aim is to investigate the association between these selected genetic polymorphisms and cholesterol-lowering effect of statin among outpatient statin users in hospital USM. This slide summarizes the methods of our study. For first part, the patient's recruitment, a total number of 229 outpatient hyperlipidemic statin users were recruited from hospital, followed by DNA extraction and SNP genotyping. For SNP genotyping, a method called PCR-FLP was performed, in which the DNA extracted was amplified and then digested using restriction endonucleases. And finally, statistical analysis was performed using SPSS software, in which recessive model analysis was used to group the patients according to minor allele carriers and non-carrier group. And pairwise comparison between these two groups was performed using Mann-Whitney test and independent t-test. This table summarizes the PCR-FLP protocol, including the primer sequences as well as restriction enzyme used for FLP reaction for all SNPs. Moving on to the results, the first is patients' demographic profiles. Looking at the table, focusing on the bottommost part of the table, the mean lipid level at baseline, you can see that our population had elevated total cholesterol and LDL level above normal range. While for HDL level, our population had reduced as compared to the normal range, while for baseline triglyceride level was borderline as compared to the optimum level. The second result is the comparison of genotypic distribution of our population and the reference population using chi-square analysis. Reference population here is referring to East Asian populations from ensemble database. As you can see in the table, focusing on the p-value of each SNPs, you can see that CETP RS708272 and COQ RS469375 was not significantly different as compared to the reference population, while for the other SNPs was significantly different. While for ApoE genes, as you can see in the table, focusing on the p-value, you can see that genotypic distribution of ApoE gene was significantly different to the reference population. And our main finding for this current study, the first is ApoE5 RS662799. As you can see in the figures, that the minor allele carriers of this SNP had higher HDL level and reduced triglyceride level as compared to non-carrier after 7 to 12 months of statin treatment. Next SNP is ABCC2 RS717620, in which you can see in the figure that the minor allele carriers had a lower triglyceride as compared to non-carrier up to 7 to 12 months of statin treatment. And finally, for ABCG2 RS22311142, you can see that the minor allele carriers had higher total cholesterol as compared to non-carrier after 7 to 12 months of statin treatment. For the discussion part, for ApoE5, based on previous study, had been associated with decreased plasma triglyceride level that is caused by reduced VLDL synthesis in the liver. And you can see in the current study, the SNP of ApoE5 RS662799 carriers had a better response to statin treatment with regards to increased HDL and reduced triglyceride level. For ABCC2 RS717620, based on previous study, had been associated with reduced gene expression, therefore causing an increased statin bioavailability and enhanced efficacy. And our current study showed that these minor allele carriers for this SNP had a better response in terms of triglyceride reduction. And finally, ABCG2 RS22311142 also had been associated with gene expression. However, our current study had shown contradicting findings, and we come up with one possible explanation that is due to drug class effect, in which based on previous study, ABCG2 had been shown to have preferences for certain statin. However, our study did not specify finding according to statin types, which therefore might give different findings as compared to previous study. As conclusion, statin efficacy is associated with genetic polymorphism, in which we found out that ABCC2 RS717620 and ApoE5 RS266279 had a better response, while for ABCG2 RS223114 had a reduced response. And for future study, we strongly recommend a future multi-centered study involving more SNPs with stricter recruitment criteria, in which will certainly help to improve understanding towards this field. That's all for my presentation. Here's a list of references, and thank you.
Thank you, Amirul. I invite the judges to ask their questions.
Okay. Hi, Amirul. Very good presentations. Congratulations on the good work that you have done as well. I would like to ask one question. I know you have recruited patients from one particular clinical center in Malaysia.
Yes.
I didn't know I'm not sure if you actually stratified them based on their ethnic groups, number one. And have there been any studies, similar studies in other parts of the world? And is there a variation between Malaysian population and the rest?
Okay, should I open the camera or, okay.
Yes, you can turn on your camera.
Okay. The first question is, may you repeat the first question? Sorry, I missed the part of the first question.
I wanted to know if, have you looked at the ethnic variation in this SNP studies that you have done? Because we do have a multi-ethnic population and you have recruited 229 or so patients. So that was my first question. Was there any ethnic variation? And are your results any different from the studies conducted in other parts of the world, in other populations?
Okay, thank you. For the first part, we did not stratify our finding according to ethnicity. Means that the 229 is majority from Malay population, but for the result, it's actually the pool one, it's not the stratified. And the second one, the question is whether is there any variations between our populations and other studies. So far, we focus on SNPs. For CETP Association, there's a previous study in nearby Thailand population in, I think, around 2018, by one Masai and colleagues.
we showed the similar findings in which the B2 allele, or you can see the minor allele, had a better response. But our finding for CTP, we did not find any significant association. For our study, we only find association in these three SNPs, which is the APOA. The APOA5 RS62799 is similar to the North Iranian population by I'm not sure the name of the researcher, but the population is from North Iranian population. While the second SNP, which was significant for our study, is RS ABCC2 7176200. That one is according to the theory, yes, it has better response, but I'm not particularly sure about the other population. But for the third SNPs, ABCG2 RS2231142, we did find a different findings according to the theory. According to the theory, the minor allele has supposed to be a better response, but our finding RS2231142 is different finding. So this might be a different finding compared to previous populations lah. But about the previous populations, I'm not particularly sure about the SNP. So I think that's my answer for the second question.
Okay, thanks, Amirul. I have no further questions.
Thank you, Dr. Prof. Amanda and Amirul. We'll move on to the next presenter. Dr. Hafiz Awais Nawaz, I hope I'm pronouncing that correctly, from UVAS, Pakistan.
Hello, greetings from Lahore, Pakistan. I am Dr. Hafiz Awais Nawaz. I have been associated as a faculty member with the Institute of Pharmaceutical Sciences, University of Veterinary and Animal Sciences, Lahore, Pakistan. So today, on behalf of my team, I would like to present on the topic of evaluation of drug-drug interactions among pediatrics in tertiary care hospitals, finding from Pakistan. So we can call the drug-drug interaction as when it occurs when the two or more drugs react with each other. Such type of reactions can be safe, but sometimes some adverse drug events based on these DDIs can lead to the severe morbidity or the mortality. So FDA reports almost 2 million type of the adverse drug events in the USA each year. So that's a huge number. And that can go up to the killing of approximately 100,000 people each year. That's a huge number. Out of these, 35 to 40% of these severe ADEs are preventable. We assume that, and that 5 to 35% are caused by only because of the drug-drug interactions. We believe that there is an opportunity to improve the health outcomes by employing the medication reconciliation process that effectively prevent these drug-drug interactions. We also believe that among many type of the reasons, polypharmacy is a big reason for these type of the drug-drug interactions. And why we have these polypharmacy available among the patients. For example, if you have an upset stomach, you go to the gastroenterologist. That pill from the gastroenterologist can ease your stomach, but that can lead to some itchy rash. You go to the dermatologist, and that pill again can ease your rash, but can cause depression or some type of the other problems. You go to the psychologist to treat those problems, but that can cause some other problems in you. So this cycle goes on. So the ultimate problem that happens afterward is the uneasiness of the patient and the less trust on the medication system. So the aims and objectives of this investigation was to identify the drug-drug interactions in hospitalized patients, and this is a specialized one for the pediatric patients. We want to also associate these drug-drug interactions with polypharmacy, many of the medication that has been co-prescribed within the same prescription. We would like also to categorize the drug-drug interactions according to the severity as a minor, moderate, or the severe type of the drug-drug interactions, also on the basis of the pharmacokinetic or pharmacodynamic type of the interaction. We also wanted to identify the patients and practices at the increased risk of the potential drug interactions in this regard. So how did we do that? It was a cross-sectional prospective analytical study, and that was performed at the Pediatric Tertiary Care Hospital in Lahore, Pakistan. The overall duration was 4 months, and almost 300 patient prescription orders were examined. Data was collected from the patient medical files about the previous and the current medication history, and the drug interactions analyzed through the interaction checker on Medscape interaction checker. We know there are many available, but we check from the Medscape. So the drug-drug interactions were also categorized on the basis of the severity levels, also on the level of the pharmacokinetics or the pharmacodynamics. So what we found initially was that most of the patients that had these type of the problems, they belong to the early in the age from the 1 month to 5 years of age. Also, only there were lesser number that belong to the 5 year to the 10 year, and also some that belong to the 10 years to the 14 years, as it is the pediatric hospital. So the maximum age limit was 14 years. So no other patients were also there. Also, the distribution of the number of the drug examined in the prescription was only there were 10.7% of the prescription that had 1 to 3 number of the drugs available within the same prescription. There was a good number, about 32.7% that had 4 to 7 number of the drugs in the same prescription, and more than 50%, 56.7% was a prescription that had more than 7 number of the drugs available within the same prescription. You can see it's a huge number that most of the prescription had high number of the drugs within the same prescription. Also, if we can say that, also we checked for the number of the drug-drug interactions available in them. Yes, there was a high number with the less number of the drug interactions, but there was a huge number that had the high number of the drug-drug interactions available in them. And as you can see that if there are one drug-drug interaction, that could lead to the problem. And if you have more number of the drug-drug interaction, that could be very unsafe for the patients. Also, we categorize them on the basis of type of the severity. So there were lesser number of the drugs that had the minor type of the interaction, but there were huge number that had the moderate to severe type of the interactions available. Here, I'm just representing some of the drug-drug interaction with the high prevalence percentages here. If you can see, the first one had the ceftriaxone interaction with the calcium gluconate. That was the severe one, could lead to the precipitation in the lungs and kidneys. That was on the basis of pharmacokinetic, but there were many who were based upon the pharmacodynamic type of the interactions, and all of these type of the DDIs that could lead to the increased toxicity of the other drug that was co-administered. So that's a good number. If I can summarize that, so moderate to severe drug-drug interactions were found, and there was a high incidence of the percentages of polypharmacy available. More number of the pharmacodynamic drug-drug interactions than pharmacokinetic type, and many interactions have been reported before. That's a dilemma. Polypharmacy has been considered as a major challenge for the DDIs, and we can suggest that the electronic prescribing can be adopted with the automated DDI checker that could lessen the number, the percentages. Appointment or the involvement of the clinical pharmacist can also lessen such type of the problems, and the continuous training of the physicians and pharmacists is recommended. So these are some of the references that I have utilized during the preparation of this presentation. I would like to acknowledge the MSPP for decorating such a good platform and providing the opportunity. I would like to thank my team, especially to my Director of the Institute of Pharmaceutical Sciences, Dr. Tahir Khan, for his motivation, and also to my university and HSE for all the things that are happening today in Pakistan in the universities. Thank you very much.
Thank you, Dr. Hafiz. I open the floor to the judges to ask any questions.
Thanks very much. I can start off. Very nice talk. I mean, clearly there was a significant number of patients that had a high number of drugs. Were they being prescribed by multiple doctors, or one doctor or pharmacist, or was it a combination of all those?
Hello, thank you very much for your question. Could you hear me clearly?
Yes.
Yeah, thank you. You know that there were like more than 300 patients available in that has been included in the study. So that's actually not possible that they have been prescribed by the same doctor. So there were many. They have been categorized in the different wards and examined by the different physicians. So that was clearly not possible that they have been prescribed by the single physician. So there were many.
Yeah, no, sorry, you misunderstood my question. So was one patient seeing one doctor, or was one patient seeing multiple doctors?
Yeah, one patient is seeing one physician. Yes.
Right. So one physician or one pharmacist is prescribing up to 7 drugs for one patient.
Correct.
Right. Okay. And so how would you potentially train or interact with those doctors to get them to reflect on whether that was appropriate, etc.?
Yeah, that's a very good question. Actually, the clinical practices has not been very established in these areas, and we have been trying to recommend that we should do that. And for that, such type of the studies are actually needed to start this. So now we did that, and you know that there are many differences between the drug metabolism in the adults and as compared to the pediatrics, and unfortunately many of these studies are on the adults, and in the pediatric they have been missing in the globe, also especially within our country. So we tried that, and also now with these studies we can discuss with the administration, also with the working force, the physicians and the clinical pharmacists there, and then recommend that these practices are not very comfortable, and we should adopt the other means to minimize these things in the future. That not only enhance the patient compliance, but also all of the problems that are happening with the patients, there could be the reasons behind are some of these practices.
Were you surprised by the overprescribing or the polypharmacy? I mean, it seems quite significant.
Yeah, I mean, I really don't understand why they have been doing this. Especially, I really understand that what happens in the older stages when there are multiple diseases with the patient, that there is aging going on, there are problems with the liver, with the kidney, with the stomach, and many others. So polypharmacy is quite understandable. But what is happening with the infants, with the small children, I really don't understand. So I mean, they just ask about or just they see the symptoms and they just start adding the medication without understanding the root cause of these ailments in them. So what's the big problem is the patient compliance. We have been trying to really talk about that, but we are still at the very early stage.
Well, thank you very much. A very interesting talk. Thank you very much.
Thank you.
I just want to ask one clarification,
Yes,
Dr. Hafiz. You know,
Yes.
This is in a hospital setting, correct?
Yep.
So the prescribing was done by different doctors or different specialists, but the dispensing at the end of the day was done at the counter, at a pharmacy counter.
Yeah.
So would pharmacists not be looking into the drug-drug interactions or probably even flag it to the clinician?
Yeah, I mean, you are very right. There should be a big force of the clinical pharmacists who should have been understanding, identifying, and trying to minimize that. But the clinical practices are at a very early stage, number one. So there is not a big force available right there, number one. Number two, when the number is very small and they are hospitalized patients and the medications are really administered at an urgency. So there will be little chances available with a little personnel and a little time that they can check all of the individuals. I mean, if you see the earliest past of that, there were like 300 patients that we have checked, and out of these, approximately in the 60% patients, we have found these interactions. So still 40% patients are really safe. Yeah, so this is good.
Okay, thank you.
Yeah, thank you.
Thank you, Dr. Hafiz and judges. We move on to the next presenter.
Thank you very much.
Okay. The next presenter is Mohammad Aizudin from the University of Technology, Mara.
Assalamualaikum and very good day. Today I would like to present my research mainly focused on neuroprotective effects of Bdnf against retinal injury and visual impairment. Glaucoma is a disease caused irreversible blindness and projected to affect more than 76 million in 2020 and 111.8 million in 2040. Studies have reported a deposition of amyloid beta in Alzheimer's disease also have a possible immunological pathway of RGC destruction. Amyloid beta accumulation and tau hyperphosphorylation causes neurodegeneration of RGC in glaucoma patient. Inhibition of amyloid beta pathway reduce glaucomatous RGC loss. In glaucoma patient, there were decreased level of Bdnf in retina cause neuronal death. Amyloid beta decrease the Bdnf mRNA expression and lead into decreased activation of TRKB receptor cause impairment of Bdnf signaling. Bdnf is a chemical produced by brain, function in dendrite growth and stimulate neurogenesis. The objective of this study aimed to investigate the neuroprotective effects of BDNF against amyloid beta 1 polypeptide, induced retinal injury and visual impairment in Sprague-Dawley rats. In this study, rats were categorized into 3 groups: control, 140 group, 140 co-injected with BDNF, and rats were subjected to 5 different parameters. First, we want to observe the effects of retinal injury using H&E staining. From this study, we found out that 140 group shows significantly lesser number of retinal nuclei as compared to BDNF and control. Further assessment were done using retrograde labeling, where we injected fluorogold staining, and we found out that in 140 group, lesser number of live retinal ganglion cells were observed as compared to BDNF and control. The apoptotic signal using tunnel staining also shown similar result, where 140 group express higher number of apoptotic signal as compared to BDNF and control. Next, we move into optic nerve damage assessment, where we found out that in 140 group, number of nuclei condensation and vacuolation were significantly higher as compared to BDNF and control. Similarly, when we observed the oxidative stress marker and BDNF concentration using ELISA, we found out that in 140 group, the activity of SOD, GSH, catalase, and BDNF were significantly lower as compared to other group. When we proceed into the object recognition test using open field arena, where first rats will undergo the familiarization stage, where they will introduce with 5 novel objects, proceed with first partial test, where object 4 and object 5 location were displaced from its original location, and object 1 were replaced with object 1A. And from this study, we found out that during familiarization stage, there are no significant difference between all groups. However, the condition changed when object 4 and object 5 were location were displaced from its original location, and it caused amyloid beta 140 group remain significantly high number of approaches to object 1, 2, and 3 as compared to other group. And when we replace object 1 with object 1A, amyloid beta 140 shows significantly high number of approaches to object 2, 3, 4, and 5. Then we proceed into Morris-Water maze, where we use this study to evaluate visual impairment in rats. And from this study, we measure the time taken of rats to find the hidden platform. And from this, we found out that 140 group show significantly longer time taken to find platform as compared to BDNF and control group. On the right side, show the trap plot of rats in Morris-Water maze. Next, we proceed into expression of Western blot and PCR. We measured TRKB, ERK1 and 2, and caspase-3. For TRKB in ERK1 and 2, 140 group shows significantly lesser expression as compared to other group, while for caspase-3, the expression of caspase-3 was significantly higher in 140 group as compared to other group. Hence, this study showed that amyloid deposition will lead into calcium influx and lead into activation of calpain. Activation of calpain will lead into cleave of the RKB receptor and lead into decreased production of expression of BDNF. Activation of calpain also will lead into dysregulation of CDK5, phosphorylation of tau, and formation of neurofibrillary tangle. At the same time, overactivation of calpain will lead into production of caspase-3. With the help of superoxide and ion, it will eventually cause neuronal death. Superoxide and ion will not be able to regulate to its normal condition due to low number of SOD, low GSH, and low catalase activities. However, with presence of BDNF, it activates the RK1 and 2 pathway and increase production of BDNF expression, and preventing activation of calpain and dysregulation of CDK5. Lead into preventing of phosphorylation of tau and formation of neurofibrillary tangle. At the same time, BDNF will regulate the calcium influx and prevent the production of caspase-3 and eventually prevent the death. Superoxide and ion will regulate with increased number of SOD, GSH, and catalase. Hence, this study did prove that 140 induced rats did develop retinal and optic nerve injury, and BDNF prevent retinal cell apoptosis causes by 140. I would like to thank my group, and for that, I would like to say thank you.
Thank you, Mohammad Aizudin. Judges, you may ask your questions.
Thanks. Very nice talk. Could you tell me a little bit more about your model? Is this something that's been used widely before, or is this something that you've developed more locally?
Before that, did you hear me clearly?
Yes, of course. Thank you.
Okay.
Yeah.
Okay. These models actually before this 140 widely used in brain. However, in retina, using intravitreal injection, they use mainly 142. But in our study, we actually use 140. So we actually optimize doses and we come out with specific doses to actually develop specified model that we actually can represent that what actually happen as acute glaucoma situation.
And is the damage to the retina and the nerve get more with time, or is it dependent?
Okay.
It's just like an all or nothing effect. So the duration of the treatment doesn't matter. Yeah.
We published our optimization study where we actually measured within 7 days, 14 days, and 30 days.
Yep.
And we found out that 14 days actually the most optimal time where the most damage can be done. However, started from day 30 up from 40 days until 30 days, maybe due to because we just injected once, so the damage is actually stopped. So there are no further damage of the retina and optic nerve observed that being published in for our optimization study. And yeah.
Thank you. And you may not know the answer to this. Can you administer the BDNF at different stages after you've started the model, or do you think you have to do it at the start, or you don't know that yet?
Okay. Based on literature, we found out that before we started to get when we actually injected BDNF, so we started to either we plan to do pre-treatment, post-treatment, or co-treatment. But then we found out that BDNF is more suitable for pre-treatment before the injury is actually being done. So that's why in this study, we injected together BDNF and amyloid. Why? Because amyloid, from our optimization study, we found out that the damage can be clearly seen after 14 days. So we give some time, but because we want to minimize the invasive injury we done on the eye, on the rat's eye, so that's why we injected once, but together.
Thank you. Thank you very much.
May I ask one simple question?
Okay.
I just want to know, I know you gave BDNF and all that, but without it, did you compare the damage to the brain cells with and without it? And what percentage of difference did you notice?
Pardon? I can't hear very clearly.
Did you compare with and without BDNF in these animal models?
Tau.
Without giving the BDNF.
Without giving the BDNF. The damage without giving the BDNF, we actually measured before during our optimization study. In this study, we want to compare what happened if we injected BDNF. Because we already done before and we observed 140 show the damage, and after 14 days, the damage is being done. They cannot recover it back. So then we proceed with this model to see the effects of BDNF. Either they actually can prevent the damage from actually happening. So
since you talked about oxidative stress in the pathophysiology of it, did you try compare with any other studies that used antioxidant treatment to treat this condition?
Actually, no, we did not compare, but then we measured the level of oxidative stress started from day 7 until day 30, as we done in our optimization phase. And in this study, we just want to show actually the effects, what actually happened once BDNF is being inserted. That's all.
Thank you. Thank you.
I hope I answer your question.
I guess so. Yes. Thanks, Aizudin.
Okay, thank you.
Thank you, Mohammad Aizudin and our judges. We move to the next presenter. Now we will hear from AIMAN from University Technology Mara.
Greetings. My name is AIMAN, representing Center for Neuroscience Research, Faculty of Medicine, UITM. Here presenting my abstract entitled Effect of Trans-resveratrol on Retinal Oxidative Stress in rats with NMDA-induced retinal excitotoxicity. Here are the outlines for my presentation today. So let's talk about glaucoma. Now, glaucoma is an ocular disease that has affected the lives of more than 76 million people worldwide. Currently, it is the leading cause for irreversible blindness in the world. It can be divided into two main subtypes, which are people with normal intraocular pressure or IOP and those with elevated IOP. However, these two subtypes are potentially to develop glaucoma due to excitotoxicity neuronal injury, which can cause by excessive influx of calcium ion that are caused by excessive synaptic glutamate. In turn, this will lead to retinal ganglion cell or RGC death that will lead to visual loss and finally leads to glaucoma itself. So let's talk about the normal physiology condition. We can see that the ion move freely from presynaptic neuron through the synaptic cleft onto the postsynaptic neuron. But in case of excitotoxicity, the excessive amount of calcium is diffusing into the postsynaptic neuron, hence triggered many apoptotic cascades. One of the main hallmark characterization of excitotoxicity is oxidative stress or OS. This happens when reactive oxygen species or ROS production overwhelms the intrinsic and oxidation capacity. In turn, it will cause damage to cellular macromolecules such as DNA, proteins, and lipids. And in time, it will cause pro-apoptotic signaling leading to glaucomatous neurodegeneration. My compound of interest is trans-resveratrol or TR. It is a polyphenolic compound. Previously, it effectively caused significant and sustained IOP reduction through adenosine A1 receptor modulation in rat model of glaucoma. It also increased the ganglion cell layer thickness and reduced the retinal oxidative stress in rat with elevated IOP. So my proposed mechanism of action for trans-resveratrol is that it will stimulate the adenosine A1 receptor to counteract NMDA-induced excitotoxicity. This in turn will lower calcium excessive influx, and this will lead to lesser apoptotic cascade triggered, hence lower oxidative stress as well. Interestingly, trans-resveratrol also have a direct effect towards reducing oxidative stress. For my problem statement, previously trans-resveratrol has been shown to effectively reduce retinal oxidative stress and IOP in rats with steroid-induced ocular hypertension. Notably, both pressure-dependent and independent loss of RGCs in glaucoma is known to involve excitotoxicity, and adenosine A1 receptor stimulation is known to reduce excitotoxic neuronal injury. Although trans-resveratrol is known to reduce IOP via stimulation of adenosine A1 receptor, it remains unclear it could provide neuroprotection by reducing retinal oxidative stress via adenosine A1 receptor stimulation against NMDA-induced excitotoxicity, independent of elevated IOP. For my general objective is to determine if trans-resveratrol provide neuroprotection by reducing retinal oxidative stress via adenosine A1 receptor stimulation in rats with NMDA-induced retinal damage. For my specific objective is that to determine if trans-resveratrol reduce retinal oxidative stress through adenosine A1 receptor stimulation in rats with NMDA-induced retinal damage by estimating retinal levels of GSH, SOD, and CAT, and to determine if trans-resveratrol protects retinal morphology via adenosine A1 receptor stimulation in rats with NMDA-induced retinal damage by examining the HNA stained retinal. As for my methodology, the rats were divided into 4 consecutive groups. Namely, Group 1 is PBS, Group 2 is NMDA 160 nanomole, Group 3 is trans-resveratrol 4 nanomole with NMDA 160 nanomole 24 hours later, Group 4 is trans-resveratrol of 4 nanomole plus DPCPX of 8 nanomole with NMDA 160 nanomole 24 hours later. Now, DPCPX is a selective adenosine A1 receptor antagonist. 7 days later, the rats were euthanized and samples collected for ELISA, which are COD, CAT, and GSH, and HNE staining as well. So here are my results. Let's go through one by one. So A represents retinal CAT. As we can see in the TR treated group here, it showed higher level of retinal CAT as compared to NMDA group, indicating higher levels of activity for the CAT antioxidant. In contrast, for the TR-DP-CPS group, it showed lesser levels of CAT, indicating lesser activity of the CAT antioxidant. Similarly with B, for the retinal GSH, the TR-treated group showed higher levels of the GSH antioxidant. This indicates higher levels of GSH activity. In contrast, the TR-DP-CPS group showed lesser levels for GSH, indicating lesser activity for the GSH antioxidant. Last but not least is C, for the retinal SOD. The TR-treated group showed higher levels of SOD compared to NMDA group, indicating that it have higher levels of the SOD antioxidant. In contrast, the TR-DP-CPS showed lesser levels of retinal SOD, indicating lesser activity of the SOD antioxidants activity. This proved that DP-CPS partially abolished the therapeutic effect of TR. In correlation with the ELISA results, I also prepared a hematoxylin and eosin representative staining for each of the group. As you can see, indicated by the red arrow is the GCL layer. This is where all the RGC resides. As you can see in A for the PBS group, there are more numbers of RGC. In contrast, B, which is the NMDA group, there are few numbers of RGC located there. In contrast with C, which is the TR-treated group, there are slightly more numbers of RGC as compared to B. Interestingly, as we can see in D, is that the number of RGC is slightly lesser as compared to C. This indicates DP-CPS partially abolished therapeutic effect of TR. So back to my proposed mechanism of action for transresveratrol. By editing DP-CPS, which is a selective adenosine A1 receptor antagonist, will partially abolish the therapeutic effect of transresveratrol. So in conclusion, intravitreal transresveratrol protects against NMDA-induced retinal oxidative stress and changes in retinal morphology in rats. This effect of transresveratrol is likely to involve stimulation of adenosine A1 receptor. Further studies to determine the precise mechanism of action are warranted. I would like to thank Center for Neuroscience Research, LACHU, IMB, and the Ministry of Higher Education for giving me the chance to do this. That is all from me. Thank you. Any question from the floor?
Thank you, Afiq Aiman. Judges, you may proceed with your questions.
I'm wondering if Prof. Prof. Fain can go first. Yeah.
Yeah, I'll just start and then you can come in, Amuda. So nice talk. Could you tell me a little bit more about your model? How much of the effects we looked at are time dependent? So you gave the MMDA 24 hours later, you waited 7 days. Is that something you've done in preliminary studies, or is that a well-established model?
Well, first of all, thank you, sir, for the question. So for the NMDA, the dose that we decided to use for the study is basically from another study that was conducted in 2018, in which the dose-dependent study for the NMDA itself. There were 3 consecutive doses that we used, 80 nanomole, 160, and 160 nanomole respectively. In the study, we observed the fact that 80 nanomole does not give the desired effect to recreate the condition in glaucoma. For the 160 nanomole, it gave the optimal condition that we are desired to see in glaucoma patient. For 320 nanomole dose of concentration, we can see that there is a sign of necrosis, and this is hardly observed in glaucoma patient. Hence, we do not use that dose. So we are sticking to the 160 nanomole dose for this study.
Thanks very much. What about the TR too? Is it the matter when you add the TR, or how dose dependent is the effect of the TR?
Oh, yeah. Thank you, sir. So for TR, previously, the dose for 4 nanomole was used in a different model of rats, which is a intracerebral hemorrhage stroke model, in which in the study was conducted in 2020, there is an effect. It managed to restore the retinal oxidative status, and also it preserved the eye structure of the rats. Therefore, we are using that particular dose of 4 nanomole to a different model, which is my model, the NMDA-induced excitotoxicity model. So we are changing the model of the rat itself.
Thank you. Amuda.
No, I have no questions for this. Good presentation, Afiq.
Thank you. Thank you.
Thanks very much. Nice presentation.
Thank you, Afiq Aiman. And thank you, Prof. Win, for your questions. We will move to the next presenter. So the next presenter is Siti Hajar Zabri from University Science Malaysia.
Hello everyone, my name is Siti Hajar Zabri and I will be presenting my research titled Characterization of the Reward Structural Connectivity in Female Malay Adolescents using Diffusion Magnetic Resonance Imaging. And my supervisor is Dr. Asma Hayati Ahmad and my co-supervisor is Associate Professor Dr. Rahimah Zakaria, and I am a master student from Department of Neurosciences, School of Medical Sciences, University Science Malaysia. So the reward network is the regions involved in processing and responding to reward stimuli. The reward structure connectivity has previously been studied, especially concerning reward sensitivity and addiction. So there were many previous studies. So this is an example of a study that I show you by Vanderbosch and colleagues in 2015. So basically they did a diffusion MRI study and chose the CN targets based on the previous studies which were consistent with the frontostriatal circuits, including primates and other human diffusion MRI studies. So they chose the striatum as seed and 10 a priori target mass, which are anterior cingulate cortex, posterior cingulate cortex, dorsal ACC, medial orbitofrontal cortex, MOFC, inferior frontal gyrus, supplementary motor area, dorsolateral prefrontal cortex, DLPFC, ventrolateral prefrontal cortex, VLPFC, and hippocampus and amygdala. So the adolescents are of the age of 10 to 25 years old based on previous brain studies. They are known to be associated with higher risk-taking behavior and impulsivity. Adolescence is also often the time of onset for substance abuse. This may be due to the constant development of brain regions during the adolescence period, and adolescents have also been shown to have higher reward sensitivity compared to other age groups. The diffusion MRI is an excellent tool for investigating the white matter integrity connecting the reward regions of the adolescent's brain. So the research questions are, what is the characteristic of the reward structural connectivity in terms of connection strength, which is obtained from the number of streamline in Malaysian Malay adolescents, and what is the characteristic of the reward structural connectivity in terms of parcellation of NACC in Malaysian Malay adolescents. So the research hypotheses are the relative connection probability and parcellation of the NACC to the 6 reward-related regions would show highest connection probability and strongest connectivity to the MOFC compared to the other 5 target regions, amygdala, ACC, hippocampus, VLPFC, and DLPFC. The parcellation of NACC would show more widely distributed pattern of connectivity to the MOFC compared to the other 5 target regions. So specifically, the study objectives are to determine the connection strength from relative connection probability of NACC, which is the seed shown here in the figure, and to amygdala, ACC, MOFC, the hippocampus, VLPFC, and DLPFC. So these are the target regions. To parcellate the NACC according to its connectivity with amygdala, ACC, MOFC, hippocampus, VLPFC, and DLPFC2, and to describe the pattern of distribution. So basically, we got the ethical approval from Japan USM, and the research design is of a cross-sectional study design. We got 15 female Malaysian Malay participants, which are adolescents, and the data was obtained from USM Health Campus, Kubang Kriang, Kelantan. And to ensure the participants had typical reward responsiveness, the reward responsiveness scale, RRS, and the behavioral inhibition system and behavioral activation system scale was given to the participants to answer via online questionnaire. So the DTI database of 15 female Malay adolescents were obtained first. You can see within the study flowchart. And we did the pre-processing of DTI data, contact participants for online questionnaire via the Google form, probabilistic tractography to obtain the connection probability, calculate the relative connection probability, parcellation analysis via the fossilized FSLIs, and statistical analysis via SPSS. Here I show you an example of graphical user interface of the FSL. Here's a more example of the interface and fossilized application that I use within the current study analysis. Here are the fossilized application. So the results are from the relative connection probability found that the highest relative connection probability of NACC is to the MOFC in both the left and right side of the brain compared to the 5 targets that I mentioned. Result of parcellation analysis showed that the NACC parcellation have the widest distribution to the connection of MOFC. You can see here represented by the beige color compared to the 5 targets in both sides of the brain. This is an example of one of the participants. So the discussion is basically the current studies showed that the highest connectivity of the NACC and MOFC compared to other targets which support the hypothesis, and this finding support that NACC and MOFC have the highest connection strength compared to the 5 targets. NACC and MOFC are 2 key nodes of reward network. The tract connecting NACC and MOFC was even distinctively characterized by Casco et al. As accumbofrontal tract. And this tract the finding of this study basically may be attributed to the early maturity of this tract. So what they found is that this tract matured quite early during mid-adolescence. They found that the average fractional anisotropy of the accumbofrontal tract undergoes significant changes with age across the lifespan, whereby there is an early peak at age around 14.8, followed by a decrease, then leveled out. There was an even sex difference that they found, whereby this tract of males matures earlier compared to the females, which is around 13.9 versus 18.6. So males mature earlier within this tract. So basically, the conclusion is that the relative probability of connectivity and parcellation showed the strongest connectivity of NACC to the MOFC in Malay adolescents, which are females, and this support the finding of highest connection strength of NACC MOFC of previous studies. Thank you.
Thank you, Siti Hajar. Judges, you may proceed.
Interesting presentation, Siti Hajar. I was going to ask why you decided to conduct this study on female Malay adolescents, but you had that very last point in the discussion saying why you did that. But I would like to know, what is the relevance of this study to life and in practice?
Okay, so basically adolescents themselves are different with other age groups. So we should look at their brains within any ethnics basically. But I got female Malay adolescents from a database which was actually collected previously. So I actually used that database and took the participants, contacted them again, got their reward responsive scale to see their reward sensitivity, reward responsiveness. And then I proceeded with looking at their brain. This is important other than what I just said. This is important because I saw many reward network studies, reward circuit studies. They actually, a lot of them relate reward and addiction or reward sensitivity with this tract, NACC and MOFC. So that's why I look at NACC as a C. Because if you look at what I said previously, this one study they used striatum as seed, but I use NACC in particular because it's very interesting that many studies use that. So yeah, and I found that it's actually very strong connection there, and it could be the same in other ethnicities as well. But I also found individual variant, so it's not the same as in I found the same thing for everyone. I actually found individual differences as well. Yeah.
How can we use these results in a clinical situation?
Clinical situation. So a couple of studies, like when they look at smoking, for example, substance abuse, they can see that's like a track, for example, strength of track is stronger and stuff like that. They can actually manipulate in terms of that. I'm not sure if they give drugs or anything, but it's like a way that they can actually trigger that can actually make the people who abuse, meaning smoke, maybe they can like lessen the smoking by what you call it, not adjust, triggering effect on this tract, matter tract, what matter tract that is different with normal human and not normal human, healthy typical people. So in comparison, they have different in tract, for example, stronger connectivity or anything. So when we find something like this, we can actually see what's the difference between them and the substance abuse adolescents, and actually trigger something that can actually help those with substance abuse. Yeah.
Thank you,
That's
Siti
actually-
Hajar.
yeah.
I think it's a very interesting and also an important study.
Okay, thank you.
Thank you.
Thanks, Siti.
Thank you, Siti Hajar. And thank you, judges. We move on to our last presenter for today, which is Rubia Kurushit from Lovely Professional University, India.
Hello everyone, myself Rubia, and I'm a research scholar from Lovely Professional University, Punjab, India. And the title for my presentation is Giving New Birth to Symbiotics as Pharmaceutical Carrier Beyond Their Use as Nutraceuticals Case Studies. Symbiotics, which are a combination of probiotics and prebiotics, are well reported for their therapeutic benefits in treatment of various diseases such as cancer, diabetes, PCOS, Alzheimer's disease, and colon cancer. These basically act by reduction of gut dysbiosis, which is caused due to majority of factors such as high fat diet, sedentary lifestyle, stress, and aging. In our research, we have explored the new role of symbiotics as solid carriers, case studies of curcumin and quercetin. Curcumin and quercetin are well reported for their therapeutic activities. These basically act by reduction of ROS, that is reactive oxygen species, and these act on different inflammatory pathways and thus reduce the inflammation and oxidative stress, which are the major pathways involved in the development of many diseases such as CVDs, then diabetes, cancer, and kidney diseases. But the challenges they face are low aqueous solubility and poor bioavailability. So in order to overcome these challenges, we have developed the self-nano-emulsifying drug delivery system of curcumin and quercetin. By developing these nets of curcumin and quercetin, we will enhance the solubility of both the drugs. And also the drugs will be protected from enzymatic degradation in the GIT. These are the SNEDs formulations for individual drugs as well as mixture of both the drugs. So here composition we have taken is oil, labrafil, capnine, sulfactant 380 and 4-sulfactant transferol B. Further, we have solidified this liquid SNEDs using the combination of solid carriers, probiotics, and Ganoderma luciata mushroom extract powder. These have been converted into solid powder by using the spray drying technology, and further, this powder has been converted into pellets. Then the droplet size, zeta potential, angle of repose, drug loading was calculated for all these batches. Here we can see that after solidification, there was not much effect on the drug loading and angle of repose, and then zeta potential or droplet size. Here we can see that in case of liquid nets as well as these nets, powder and pellets, after spray drying, the droplet size is almost same. And then zeta potential has been increased after solidification. Angle of repose has been improved after spray drying and conversion into pellets. Drug loading is more than 95% in all the cases. These results show that the integrity of SNEDs is maintained after conversion into pellets. Then these are the same images of excipients as well as the formulations. This is for curcumin, quercetin, glep, probiotic, aerosol, and then non-spray dried powder. This is for non-spray dried powder, which is agglomerated in agglomerated form. But after spray drying, this agglomerated form has been converted into spherical pellets. Then from drug release profile, we can see for both the cases, quercetin and curcumin, the drug release has been enhanced. In case of raw forms, the drug release was less than 20% in 60 minutes, whereas in case of formulations, the drug release was more than 90% in first 5 minutes. Similarly, the permeability has also been enhanced after conversion into SNEDS formulations. Then this is the summary of our work. We have taken curcumin and quercetin, which are facing the challenges of low aqueous solubility and poor bioavailability. Then we have converted them into L-SNEDs, that is liquid SNEDs, to increase solubility and bioavailability. Further, these L-SNEDs face the instability challenges due to phase separation and drug precipitation with time. So in order to overcome this challenge, we have solidified it using Ganoderma Ganoderma mushroom extract powder and probiotics. Here we haven't used the commonly used carriers that are lactose and aerosol, since they face their own challenges. Lactose, it is used in very higher amounts, and aerosol is reported for silica toxicity if used in higher amounts. So in our formulation, these probiotics and prebiotics mixture, that is symbiotics, will serve a dual role as an excipient, that is as a solid carrier, as well as it will offer therapeutic benefits also, since these mushroom polysaccharides and probiotics are well reported for their therapeutic benefits. So this is the conclusion for our study. The developed formulation has utilized easily available excipients in their safer concentration, indicated the safety of formulation on long-term use. Further, this L-SNED concentrate, it can be formulated at larger scale, and it requires simple blenders for formulation. Therefore, it can be brought to a larger scale using the well-known technologies such as spray drying and extrusion sterilization, which are commonly used. Now, this research has also given a future direction to the scientists working in the field of nutraceuticals to explore the role of symbiotics as excipients for formulating other drug delivery systems. Thank you.
Thank you, Rubiya, for the presentation. I open the floor to the judges for any questions.
Thank you. I'm happy to start. So thank you. Nice talk. The SNEDs, liquid, powder, and pellets all come up with the same permeability, the same drug concentration. Is that correct? Yeah.
So can you please repeat your question?
Yeah. So with your graphs, when you use the liquid SNEDs, the powder, and the pellets, it affects the active compounds the same way, so it has the same permeability and the same drug release. Is that correct?
Yes, sir. As per graphs, queen can be used up to 1.5 ml per day. So in our formulation, we have used the concentration of queen was only 40%. That is 40 microliter in 1 ml. 40 ml microliter of queen 80 was used.
Yes.
At a safer concentration. It was used at a safer concentration, sir.
Okay. And so can you test this model with a pharmacokinetic profile, like testing drug concentrations in different components in rats or something? Is that something we can do?
Yes, sir. We have carried our in vivo study in rats. So we have carried the pharmacokinetic studies also. In case of pharmacokinetic study, we have seen that the bioavailability was enhanced to 80%, more than 80% in all the formulations, liquid SNEDs, then solidified forms also. And then we have seen the effect on diabetic rats also. We have seen that the formulation of curcumin, quercetin, and including probiotics, it was able to reduce the blood glucose level from 400 mg per deciliter to less than 100 mg per deciliter. Further, we have seen that these group, which was treated with symbiotics alone, only symbiotics, it was also able to reduce the blood glucose level and hyperlipidemia to some extent.
And you finished off on this note. So you explored the wider applicability of this technique. So what other things do you think it might be useful for?
Sir, can you repeat your question, please?
Yeah, the methodology you use with the SNEDs, in the end, you actually suggest that it might have broader applicability or might have broader uses. Could you think of other things that this might be useful for?
Yes, sir. Actually, the formulation of SNEDs is very easy procedure. It doesn't require any sophisticated instrumentation. We only require a blender. Then for solidification, we require a spray dryer, and then we require for conversion into pellets, we only require extrusion spherizer. So this SNEDs formulation, it can be commercialized very easily. And then addition of probiotics and prebiotics, our main focus was that to check the role of probiotics and prebiotics as solid carriers. So these probiotics and prebiotics have proved very good carriers in our formulation. We have obtained the powder with good flow characteristics.
Okay, thank you very much. Very interesting.
Thank you.
I have no questions. Thank you.
Thank you, Rubia, and thank you, Prof. for the questions. We have come to the end of our this session, basically. So join me again to thank Prof. and Prof. Amuda for judging this session, as well as congratulate to all the presenters for a job well done. Before we end the session, I would just like everyone to turn on their cameras so we could take a commemorative picture. One, two, three, smile. All right, thank you. So we will end this, So we will end this session, and we will join back at 4:15. Thank you, everyone.
Nice talks, everyone. Good luck.
Thanks, Alina. Good job, presenters.
Thanks, Prof. Thanks, Dr. Muda.